Good morning. I am Bruce Chabner from Massachusetts General Hospital and Harvard Medical School, and I'd like to welcome you to the latest in the series of colloquium talks on translational oncology. I'd like to thank the sponsors of this series, Novartis, Sanofi, and Regeneron, and our exhibitors. And I would like to turn this over now to our moderator, Aaron Hata, who is the head of lung cancer research at Mass General, and he will introduce our speakers. And thank you for your attendance.
Welcome, everyone. Thank you for joining us for the Chabner Colloquium MAP series. My name is Aaron Hata from Massachusetts General Hospital, and I will serve as a moderator for today's session, which includes presentations by Dr. Alice Shaw and Dr. Rebecca Heist. Before we begin, I would like to start by thanking Novartis Pharmaceuticals and Regeneron Pharmaceuticals and Sanofi Genzyme for providing educational grant support for this series. I also want to acknowledge Dr. Aditya Bardia, MAP series co-chair, and Massachusetts General Hospital for collaborating with Dr. Chabner and STO on the development of this series. We are especially grateful to the presenters for their participation. Additionally, I encourage you to visit our exhibit hall, which you can access in the timeline on the main event page, to learn more about each of our generous supporters. They include AstraZeneca, Foundation Medicine, Genentech, Novartis, Pfizer, and Takeda. They have posted informational brochures and videos for you to download and view at your discretion. In addition, you may set up a chat with a representative if you desire. Information was emailed to attendees with details about how to receive continuing education credit for this event. CME credit is available only for the second presentation today. And now I would like to introduce Dr. Shaw presenting on targeting ALK, ROS, and other fusion drivers, and Dr. Heist, who will discuss targeting MET. Immediately following the presentations, we will host a 15 to 20-minute Q&A. If you would like to submit a question to be addressed, please use the chat function of this platform to post your inquiry. Questions can be submitted anytime during the presentations. Please note that the microphones for all attendees are automatically muted. And now, without further comments, let us proceed with today's session.
Hello. Thanks to the organizers, especially Bruce, for inviting me to speak in this MAP series. Today I was asked to review targeting ALK, ROS1, and other fusion drivers. In lung cancer, these subsets have really helped to establish the paradigm of personalized medicine and have also revealed fundamental insights into drug resistance, which have informed drug development. So I'm really happy to be here to review the latest data and to address optimal management of these cancers. Now, most of the preclinical work I'm going to highlight today was done while I was still at Mass General, but as you can see here, I'm currently employed by Novartis, where I lead oncology early clinical development. And because of this affiliation with Novartis, this presentation, I'm sorry to say, is not available for CME credit. Here are my 3 learning objectives, which I think are fairly self-evident. So all of you are familiar by now with the numerous oncogenic drivers that have been identified and validated as targets in non-small cell lung cancer, starting, of course, with activating EGFR mutations and now including KRAS G12C, ALK, ROS1, MET, and many others. Shown here are just the 8 drivers for which we have approved or soon to be approved targeted therapies. But there are other targets as well, including HER2 and NRG, just to name a few. But for today, in the interest of time, I'll be focusing on one of my major research areas when I was at MGH. And this, of course, is ALK rearranged or ALK positive lung cancers. But it is important to note that many of the learnings from targeting ALK are directly relevant to ROS1 and other oncogenic drivers. So for ALK, the therapeutic landscape has evolved incredibly rapidly the last decade. Since the initial accelerated approval of the first generation ALK ROS1 inhibitor crizotinib in 2011, 3 more potent second generation ALK inhibitors, ceritinib, alectinib, and brigatinib, and 1 highly potent and CNS penetrant third generation ALK inhibitor, lorlatinib, have received accelerated approvals. Now, for these 4 next-generation ALK inhibitors, approval was initially in the resistant setting, but all 4 have now received approval as first-line therapies for patients with newly diagnosed metastatic ALK-positive lung cancer. So with 5 approved ALK inhibitors, the obvious question facing clinicians now is how to select and sequence ALK inhibitors in patients. So shown here is the original treatment strategy that evolved after the initial approval of crizotinib, which consists essentially of treating patients with increasingly potent, mutant-selective, and CNS-penetrant targeted therapies. So first-gen crizotinib, followed by a second-gen TKI, followed by third-gen lorlatinib. Starting in 2017, this sequential approach was further refined based on the positive results of randomized phase 3 studies comparing more potent second-gen TKIs versus crizotinib as first-line therapy, leading to replacement of crizotinib with second-gen TKIs, followed by lorlatinib as the standard approach. But most recently, we now have data from a randomized phase 3 study of lorlatinib versus crizotinib, showing an even greater benefit of lorlatinib over crizotinib, leading to the recent full approval of lorlatinib as first-line treatment. So now, the question is whether to continue the sequential TKI approach of second-gen followed by third-gen TKI, or to use our most active agent, the third-gen TKI, as first-line therapy. And this is the main question that I would like to focus on today, taking into account both mechanistic studies on TKI resistance, as well as the latest data from the clinic. Now, this may seem like a narrow topic, but this question is actually highly relevant to ROS1 and, in fact, to every oncogene-addicted cancer, especially as we learn more about resistance and we develop new and improved next-generation kinase inhibitors. So let's start with the sequential TKI approach and the data that support this strategy. Shown here are data from the global phase 3 ALEX study comparing first-line alectinib versus crizotinib in ALK-positive lung cancer. As you can see, compared to crizotinib, alectinib significantly prolonged PFS, with median PFS almost 26 months versus 10.4 months with crizotinib based on independent review, and hazard ratio for disease progression or death was 0.50. Similar findings have also been reported with other second-gen TKIs, and all of these data have supported replacing crizotinib with more potent and CNS-penetrant second-gen inhibitors. However, while the benefit of first-line second-gen TKIs is impressive, patients still develop resistance. At Mass General, my former group leveraged our repeat biopsy program to identify clinically relevant mechanisms of resistance to second-gen TKIs. Shown here are early data that Justin Gaynor published now about 5 years ago, highlighting the secondary mutations in the ALK tyrosine kinase domain that emerge in patients treated with first or second-gen ALK TKIs. As expected, the more potent second-gen TKIs do lead to more on-target resistance, with over 50% of resistant cases harboring secondary ALK resistance mutation. And by far, the most common resistance mutation seen is the so-called solvent front ALK G1202R mutation, which is shown here in red. Fortunately, as data were emerging on ALK resistance mutations, chemists at Pfizer were working on developing a third-gen ALK TKI, lorlatinib, that could overcome all of these mutations and also penetrate the blood barrier, given the propensity of ALK-positive disease to spread to the CNS. So shown here is a very high-level comparison of lorlatinib versus crizotinib, highlighting lorlatinib's greater potency against ALK, its cellular activity in cell line models harboring ALK resistance mutations, particularly the G1202R, and also its much lower MDR efflux ratio, suggesting high brain availability. In the lab, we confirmed both the greater potency and the pan-inhibitory activity of lorlatinib relative to earlier generation TKIs using BAF3 models. Here, first, second, and third gen TKIs were tested against a panel of different BAF3 models harboring different single ALK resistance mutations, as you can see on the left. Consistent with its design and the early biochemical and cellular characterization by Pfizer, we found that in contrast to other TKIs, which lose activity against select mutations. Lorlatinib retains activity against all known single ALK resistance mutations. So these data provided the rationale for phase 1 testing of lorlatinib with an initial focus on patients relapsing on first and second gen TKIs. So here are efficacy data from our phase 1/2 study of lorlatinib in patients who had failed one or more second gen ALK TKIs. And as you can see here, lorlatinib was quite active in this heavily pretreated population of patients with a response rate of about 40% and median PFS of about 7 months. We went on to show that, consistent with its mechanism of action, lorlatinib is most active in those patients who have failed a second-gen TKI due to a secondary ALK resistance mutation. Here's a very similar waterfall plot, but now color-coded according to ALK mutation status at the time of enrolling on lorlatinib. As shown by the blue bars, those patients with ALK resistance mutations derived much more benefit from lorlatinib than those without. The response rate was 69% versus 31%, and median PFS, which isn't shown here, was also significantly longer in mutation-positive patients. So these findings and others helped establish the sequential TKI approach for ALK-positive lung cancer with second-gen followed by third-gen inhibitors. However, while many patients have done extraordinarily well with sequential TKIs, we almost always see resistance emerge on lorlatinib. And now resistance in a patient who has failed first a second-gen TKI followed by lorlatinib is much more complex than patients who have failed just a second-gen TKI. So shown here are updated data from Jess Lin at Mass General, highlighting ALK resistance mutations identified in patients failing on alectinib or failing on lorlatinib after having received alectinib or another second-gen TKI. For alectinib, you can see on the left that most on-target resistance is mediated by single ALK resistance mutations. In contrast, when patients fail sequential alectinib and lorlatinib, as shown on the right, now there's a much more diverse set of ALK mutations that emerge, many different double and even triple ALK mutations, which suggests a sequential acquisition of resistance mutations. Now, to formally assess this, we've examined serial repeat biopsies in patients treated with sequential ALK TKIs and performed not just the typical targeted NGS, but whole exome sequencing and clonal analysis with the help of Mike Lawrence and his team at Mass General. Shown here is one example. This patient had received first-line crizotinib, followed by second-line alectinib, followed by third-line lorlatinib, each time responding, but then eventually relapsing. As you can see here, we could detect an ALK G1202R resistant subclone, and that's shown in green, that first arose on alectinib, and then under the selective pressure of lorlatinib, acquired a second ALK mutation, L1196M, in cis, leading to a G1202R L1196M compound mutant, which I've shown here in yellow, and this led to the patient's relapse on lorlatinib. Now, this is just one of the many compound mutations we've observed in the clinic, and but in general, we have seen the same pattern of stepwise accumulation of ALK mutations in many other patients. And what's worrisome is that the majority of these compound ALK mutations are not only refractory to lorlatinib, but also to all currently available ALK inhibitors. So here's data with the same G1202R L1196M compound mutant showing resistance of this compound mutant to lorlatinib, as well as to all first and second gen TKIs. Now, the discovery of these lorlatinib-resistant compound ALK mutations has fueled the search for a potentially fourth generation class of ALK TKIs, which have activity against at least a subset of these compound mutations. So shown here is recent data presented at AACR by Nuvalent, showing that their ALK inhibitor, NUV655, is highly potent against non-mutated ALK, the single mutant G1202R, and 3 different G1202R double mutants, including G1202R L1196M. Turning Point Therapeutics has also presented data at last year's AACR on their 4th gen ALK TKI, TPX0131, which has just entered the clinic. Now, based on many lines of evidence, we would predict that tumors that have acquired compound ALK mutations after sequential ALK TKIs are still ALK dependent and hence sensitive to ALK inhibition. So some patients, I think, will benefit from 4th gen ALK TKIs. However, we've now seen in the clinic that with each successive generation of ALK TKIs, there are fewer tumors that remain ALK dependent and more tumors that have developed ALK independent resistance. In fact, based on our series of lorlatinib resistant biopsies, only about 1/4 of patients have compound ALK mutations and likely remain ALK dependent. And given the diversity of compound mutations, it's only a fraction of this quarter that will be targetable with a 4th-gen ALK TKI. So we are chasing a very small population of ALK patients with specific compound ALK mutations. Now, rather than perpetuate what feels a little bit like a game of whack-a-mole with sequential TKIs, I think a better approach may be to start with a pan-inhibitory TKI, such as lorlatinib. In theory, upfront treatment should prevent the ALK growth or development of single ALK resistance mutations, which are, of course, the necessary substrate for compound ALK mutations. And in support of this, Satoshi Yoda in the lab carried out an ENU mutagenesis screen using BAF3 cells expressing EML4 ALK, and he found that the mutagenesis generated numerous crizotinib-resistant clones, but no lorlatinib-resistant clones harboring single ALK mutations. And so taken together, all of the data suggests that moving lorlatinib upfront may be able to prevent on-target resistance, and in doing so, significantly improve clinical outcomes. So how does lorlatinib perform in the clinic as first-line therapy? Well, we had a hint of data on first-line lorlatinib from the phase 2 expansion cohort of ALK TKI naive patients. And as you can see here, the response rate with first-line lorlatinib was extremely high at 90%, and median PFS was not reached. We now also have phase 3 data comparing lorlatinib head-to-head with crizotinib as first-line therapy in ALK-positive lung cancer. This is the CROWN study, which we published in the New England Journal of Medicine end of last year. And here's the study design, which is fairly straightforward. Almost 300 patients with newly diagnosed metastatic ALK-positive lung cancer were randomized to either first-line lorlatinib or crizotinib. And the primary endpoint here was progression-free survival with standard secondary endpoints, as you can see here. Now, I should note that crizotinib was selected as the comparator because at the time the study was planned in 2016 and then initiated in 2017, crizotinib was still the standard first-line therapy for metastatic ALK-positive lung cancer, with alectinib not receiving approval as first-line therapy until the end of 2017. So not surprisingly, the CROWN study was hugely positive in favor of lorlatinib. Shown here are progression-free survival curves as assessed by either blinded independent review on the left or by investigators on the right. And in both cases, the median PFS with lorlatinib was not reached, while median PFS with crizotinib was as expected in the 9 to 10 month range. As you can see here, the hazard ratios were 0.28 and 0.21, corresponding to a 72 to 79% reduction in the risk of progression or death with lorlatinib compared to crizotinib. Now, by comparison, the hazard ratios with second-gen ALK TKIs versus crizotinib have all been in the range of 0.5, so a 50% reduction in the risk of progression or death, suggesting a greater magnitude of benefit with lorlatinib versus second-gen TKIs relative to crizotinib. Now, a very important secondary endpoint in CROWN was intracranial efficacy. As I mentioned earlier, ALK-positive patients are at high risk of CNS metastases. As shown here in CROWN, lorlatinib induced CNS responses in over 80% of patients with measurable brain mets at baseline, and notably induced complete responses in brain mets in 71% of patients. And this is incredibly high compared to second-gen ALK inhibitors, which in the frontline setting have intracranial CR rates ranging from 28% with brigatinib to 38% with alectinib. Now, consistent with the high response rate, we found that time to CNS progression was also significantly longer with lorlatinib than with crizotinib. At 12 months, the CNS progression-free rate was 96% with lorlatinib, as compared with 60% with crizotinib, and hazard ratio was 0.07. As you can see here, the CNS progression-free curve with lorlatinib is almost a flat line, suggesting that lorlatinib is not only effective at treating brain mets, but can also prevent the development of brain mets. And I think this effect is likely a major contributor to the prolonged PFS seen with first-line lorlatinib. So this brings me back to the question I posed at the beginning of my talk. ALK TKIs with second and third gen TKIs versus upfront treatment with the third-gen TKI lorlatinib. I know that many oncologists may prefer sequential TKIs and knowing that there's a backup option, but based on the biology of these cancers and on all the preclinical and clinical data that we've reviewed, I think moving our most potent pan-inhibitory and CNS penetrant TKI into the frontline setting will provide the greatest clinical benefit to patients. By maximally inhibiting the target, by treating any pre-existing resistant ALK mutant subclones, by preventing the development of single or compound ALK mutations, and by treating and protecting the CNS, which we know can be a sanctuary site even with second-gen TKIs. And so for these reasons, I would vote for frontline lorlatinib. However, this isn't the end of the story because all of us expect that despite the much more durable responses and the prevention of CNS metastases with lorlatinib, patients are still going to develop resistance. Now, earlier I showed you the latest data that we have on lorlatinib resistance in the setting of sequential TKIs with many different compound ALK mutations, but overall, a majority of patients with likely ALK-independent mechanisms of resistance. Right now, we have very little data on resistance to first-line lorlatinib, but assuming we can effectively prevent and overcome single ALK mutations, a reasonable expectation is that the vast majority of patients will relapse on first-line lorlatinib due to ALK-independent resistance, which for me really highlights the urgent need to understand this type of resistance and to develop rational combination strategies. And so for the last few minutes, I just wanted to highlight some of the combination strategies that are being developed to address ALK-independent or off-target resistance. Shown here is a very high-level summary of some of the bypass or alternative signaling pathways that have been reported as mediating off-target resistance to various ALK inhibitors. As you can see, a number of different RTKs, such as EGFR, c-KIT, and MET, can be aberrantly activated to drive resistance. Also, several studies have implicated the non-receptor tyrosine kinase SRC in ALK TKI resistance. And other studies have identified specific alterations leading to activation of the RAS-MAP kinase pathway, including KRAS amplification and MEK activating mutations. In the interest of time, I'm just going to touch on a few combinations that have entered the clinic. The first is the combination of ALK and MEK inhibitors. A number of studies have suggested that this combination may be effective not only in overcoming resistance, but also potentially preventing or at least significantly delaying resistance. These studies were published some time ago, so I won't review them today, but based on these results, there are now 4 different ALK TKI MEK inhibitor combinations that are in the clinic as listed in this table. And hopefully we'll learn soon about safety and preliminary efficacy. Now, one of the combinations in the bottom row, lorlatinib plus binimetinib, is part of a larger combination study, an investigator-initiated trial that I worked on with Ibi Dagogo-Jack at Mass General, and it's worth highlighting here. In this study, patients who have become resistant to ALK TKIs without evidence of an on-target ALK resistance mutation undergo repeat biopsy and tumor genotyping, and patients whose tumors harbor MET amplification will receive a combination of ALK and MET TKIs, in this case, lorlatinib and crizotinib for its MET activity. While those patients without MET will be randomized to receive either lorlatinib with the MEK inhibitor binimetinib or lorlatinib with the SHP2 inhibitor TNO155. So why SHP2? Well, SHP2 is a tyrosine phosphatase that links upstream activated RTKs to the RAS-MAP kinase pathway and specifically mediates GTP loading of RAS. A few years back, we published the results of a pooled shRNA dropout screen in a panel of 7 resistant ALK-positive cell lines. And in this screen, SHP2 depletion was found to resensitize 5 of the 7 resistant lines to ALK inhibition, as shown in the first column of this table. These results were subsequently validated in biochemical studies and also in PDX models of resistant ALK-positive lung cancer, which showed combination benefit with ceritinib and the tool SHP2 inhibitor SHP099. Based on these results, the Mass General team has just started testing the combination of lorlatinib and TNO155 in resistant patients. In addition, I should note that Pfizer has also recently started a multi-arm study, which includes a combination of lorlatinib with their SHP2 inhibitor from Array. So hopefully we'll have data soon on whether concurrent ALK and SHP2 inhibition can overcome at least some bypass mechanisms of resistance. The last combination I wanted to mention is the combination of ALK and YAP inhibitors. There are recent data suggesting that targeting YAP could overcome ALK TKI resistance, But I think the more interesting angle with YAP has to do with its potential role in enabling survival of drug-tolerant or persister cells after initial TKI treatments. Now, in work published last year, Tsuji and colleagues showed that after exposure to alectinib, cancer cells activate YAP, which then mediates expression of MCL1 and BCL-XL, and this allows cells to survive despite the TKI. And this group went on to show that alectinib combined with verteporfin, which inhibits YAP among other targets, is more effective than alectinib alone in sensitive ALK models, suggesting potential benefit in the clinic of co-targeting ALK and YAP. Several more specific YAP inhibitors are currently under development, and so I think there's definitely potential that we'll be able to bring an ALK-YAP combination into the clinic soon. And so in summary, based on all of the available preclinical and clinical data, I would argue that for ALK as well as other oncogene-addicted lung cancers, we should be using our most potent TKI with the broadest coverage of resistance mutations and the most potent intracranial activity as first-line therapy. I think this approach will offer patients much more durable first remissions and much better control and prevention of CNS disease, which is hugely important for these patients. However, all of the data also indicate that resistance will still ultimately develop to a TKI given as monotherapy. And so the development of combination strategies that address the most common bypass mechanisms is really critically important. Finally, thinking even further into the future, I think for optimal management, we need to be more aggressive in the frontline setting, not only using combinations upfront, but also incorporating other strategies to eradicate residual disease. I highlighted one combination with ALK and YAP inhibitors, but we should also think about incorporating other distinct or orthogonal approaches, such as targeted radiation to residual disease or immune-based approaches that activate the immune system to eliminate residual disease. And so with that, I just wanted to acknowledge and thank the incredible team at Mass General, who I was so lucky to work with for over 12 years, and who have continued to lead the field with their amazing scientific discoveries and translational work. And I wanted to thank everyone here for their attention. Thanks.
Thank you to Dr. Chapner and Dr. Bardia for the invitation to talk today on targeting MET. Here are my disclosures. So today's learning objectives are threefold. First, to understand MET exon 14 skipping and amplification as biomarkers in lung cancer. Second, to review the currently available evidence for MET TKIs in the treatment of MET-altered lung cancers. And finally, to assess the current knowledge of resistance mechanisms to MET-directed therapy. MET alterations comprise about 4% of lung cancer, and there have been marked advances in the treatment of patients in this setting. As of February of this year, we have 2 drugs with accelerated approval for the treatment of advanced lung cancer with MET exon 14 skipping, capivatinib and tepotinib. And today, I'll talk a bit about the biology of MET in lung cancer, some of the TKIs that have activity, as well as emerging data regarding resistance mechanisms. MET exon 14 skipping is a driver oncogenic event in lung cancer. Mutations at the splice sites of exon 14 that lead to alternative splicing of MET was recognized many years ago, but these mutations were actually quite hard to find as they were in the intronic segments of the gene. As sequencing technologies improved, multiple groups reported on the prevalence of MET exon 14 skipping and mutations in lung cancer. And as you can see from this figure, there are a multitude of different events at the DNA level that all lead to alternative splicing and skipping of exon 14 in the transcribed mRNA and protein. The MET receptor with the deleted exon 14 loses a binding site for Sybil, and that's important for regulation of degradation of the MET receptor off the cell membrane. Without that binding site, there's decreased degradation of the receptor off the membrane and prolonged signaling and activation. These MET skipping mutations are generally exclusive of other driver alterations, suggesting that's the main driver in cancers that have this alteration. Clinically, it's important to remember that these MET skipping mutations occur at a median age that's higher than what we see for many of our other driver alterations in lung cancer. And although never smokers are disproportionately represented among those with MET skipping, this can be found across all smoking histories, including current and former smokers. In addition, it's found across all histologies, so not just adenocarcinoma, but also in squamous and sarcomatoid histologies. MET gene copy number increase can also lead to activation of the MET pathway. It's worth just taking a moment to note that polysomy and amplification are different events. With polysomy, there are multiple copies of chromosome 7 where MET is located, and that leads to an increase in the gene copy number. The same gene copy number increase, however, can be due to a different kind of event, which is focal amplification, where there's an increase in the number of copies of the MET gene without an increase in the chromosome. It's only by measuring the gene to centromere ratio that you really can differentiate between these two types of events. And here on the left-hand side of this figure, you can see that the MET to Sep7 ratio is 1, even though the gene copy number is 4. And on the right-hand side, the MET to Sep7 ratio is actually 4, where the gene copy number is 4. This is important because focal MET amplification is more likely to lead to oncogenic MET addiction than polysomy. And indeed, if you look at the presence of co-occurring oncogenic drivers in this bar graph on the right, you can see that it's really only when you have high-level focal MET amplification as measured by a MET to Sep7 ratio that you see the absence of other drivers, suggesting that this is the change in MET that's truly acting as a driver. True de novo high-level MET amplification is defined by a MET to Sep7 ratio of at least 5 as a rare event in lung cancer. These are data gathered from screening done for the crizotinib study, and high-level MET amplification was less than 1% of those screened. In vitro, multiple small molecule tyrosine kinase inhibitors have shown activity against MET skipping. Type 1 and type 2 inhibitors differ by the way they bind to the ATP binding pocket of MET, and this will be important when we think about resistance. In general, the type 1b MET TKIs are the more selective MET inhibitors, while type 2 are multi-targeted. And in the next few slides, I'm going to talk about some of the clinical studies that have shown activity of the type 1 inhibitors in MET-altered lung cancers. In Profile 101, this was an extension of the crizotinib study, the original crizotinib study, which, as you recall, was a phase 1 study with multiple extension arms. And in this study, in this cohort, patients with MET skipping with advanced disease were treated with crizotinib. All patients had to have advanced disease and no prior MET inhibitors, as is the case with all of the studies I'm going to discuss today. Crizotinib showed an overall response rate of 32% in patients with MET skipping, and the median PFS was reported at 7.3 months. Analysis of the type of alteration that led to the MET skipping event did not show a clear correlation with response. In a cohort with MET amplification as defined by MET to Sep7 ratio, the response rate was 40%. Tepotinib is another potent and selective MET inhibitor, and in the VISION study, patients with advanced lung cancer with MET skipping captured either on tissue or plasma were treated with tepotinib orally. The overall response rate in patients with MET skipping was 46%, with a median PFS of 8.5 months. And in the latest update from World Lung this year, responses in the first line and previously treated settings were similar at around 45%. Capmatinib, another potent and selective MET inhibitor, was studied in the Geometry Mono 1 study. This was a multi-arm study that treated patients with various degrees of MET amplification or MET skipping with capmatinib, and patients were placed in the different cohorts defined by the type of alteration as well as the number of lines of prior therapy. In this study, among patients with MET skipping, response rates were 41% in the previously treated cohort. So these were patients who had received chemo and or IO in prior lines. And in the first line cohort, the response rate was 68%. The median PFS was 5.4 months and 12.4 months, respectively. And looking at the amplified subsets, as measured by gene copy number here, in those with high gene copy number of at least 10, capmatinib had a response rate of 29% in previously treated patients and 40% in first line. And savolitinib is another potent and selective MET type 1 inhibitor that's been studied in patients with MET skipping. And here you see a response rate of 43% with a median PFS of almost 7 months. This chart just shows an overview of the various studies that we talked about for MET skipping. And in general, you can see that across all these studies, the median age for the MET skipping cohorts was in the early 70s. This is consistent with the median age that we see in this population. The response rates were generally similar across the board in the 40% range, with the one outlier of 68% in the first-line cohort with capmatinib. And more patients are being enrolled in an expansion cohort in that particular study to see if this holds up with a larger number of patients. Interestingly, the median durations of response in PFS, while good, are not quite the numbers we've come to expect to see based on other oncogenic drivers such as EGFR or ALK, suggesting that as a driver, MET is acting a bit differently than these other alterations. When you look at the AE profiles of the selective MET inhibitors, you see there are some class effects. Peripheral edema is quite common, and although primarily low grade, it can significantly impact people's quality of life. And GI toxicity such as nausea can be seen, as well as LFT changes. As with all targeted agents, we know that resistance to MET inhibition develops. Secondary mutations in the MET gene have been reported, and these are most commonly at positions 1228 and 1230. And these mutations are thought to interfere with the ability of the type 1 inhibitors to bind effectively in the ATP binding pocket. Theoretically, type 2 inhibitors should be able to overcome some of these secondary mutations. And here you can see in these in vitro studies that although the type 1 inhibitors did not have activity against 1228 and 1230 mutations, the type 2 inhibitors do retain activity there. However, on-target resistance mechanisms are not the only ones described. And here in this example, you can see a patient who initially responded to crizotinib and then had resistance, and at the time of resistance was found to have amplification in KRAS. And indeed, to the right, you can see this review of multiple cases put together by Recondo and colleagues. And you see both secondary mutations within MET, the on-target mutations, as well as off-target pathway activation, with activation in RAS and MET kinase pathways and ERB pathways. Effectively targeting resistance mechanisms in this setting will likely require some combinatorial strategies. And similarly here, these are pre- and post-capmatinib samples taken on patients who were enrolled in a single institution study of capmatinib. This was among patients who had actually previously received a MET TKI, unlike the other studies that I had talked about before. And a similar spectrum of resistance alterations is seen here, secondary mutations in MET, as well as activation of other bypass tract signaling, such as MET kinase and ERB pathways. Other modalities of targeting MET are also being investigated. These include MET-directed antibodies and antibody-drug conjugates. And here is a list of some of the clinical trials of these various agents that are in development and actively being investigated now. So in summary, MET exon 14 is a targetable alteration in lung cancer. MET amplification is a target as well, and how much of an oncogenic driver it is probably a bit dependent on the level of amplification. Resistance mechanisms encompass both on-target and off-target effect mechanisms in this setting. And we do have many clinical options for MET-altered lung cancer patients. These include capmatinib and tepotinib, which are FDA approved for MET exon skipping. Other type 1 inhibitors have been shown to have activity as well, and type 2 inhibitors are being investigated as well. And as I mentioned just previously, antibodies and antibody-drug conjugates are in active development. So thank you for your attention, and I'm happy to take any questions.
All right, I'd like to thank Dr. Shah and Dr. Heist for joining us for this question and answer session. I see that questions are starting to come in, so take this opportunity to enter your question into the chat for both Dr. Shah and Dr. Heist. So I'll start off by the first question that's been posed, and this is to Dr. Shah from Joe Lenners. So he is asking about ligand secretion as a potential resistance mechanism. And he is wondering how you, Dr. Shah, envision tackling these mechanisms both experimentally and eventually diagnostically.
Thanks, Erin, and thanks, Joe, for that question. It's actually a very complicated question, and I think that over the years we actually have, as you know, been studying, as I mentioned, resistance in the lab and also from clinical specimens. And what we had seen even early on was that it was not simply overexpression or activation of a receptor, for example, EGFR. It was often both upregulation of a receptor like EGFR as well as evidence of increased EGF ligand expression. So, for example, for ALK, we had seen and shown in both the lab as well as in clinical specimens evidence of EGFR activation due to upregulation of the receptor, as well as evidence of increased NRG1 expression. And so we really felt like EGFR, it was not simply a mutation or an amplification event that was activating the receptor. It was a combination of both receptor upregulation and ligand upregulation. And actually, similarly for c-KIT as well, we showed that there actually was evidence of amplification of KIT as a resistance mechanism. But also we were able to detect increased expression of the KIT ligand stem cell factor SCF as well. So again, very complicated. And I think, as you're hinting at, quite challenging for us both diagnostically as well as therapeutically. From a therapeutic standpoint, I think it is hard to imagine many different RTKs and their ligands being upregulated in the setting of resistance, and how do we target those? And that's really, I think, fueled us to think about broader approaches that I highlighted during the talk, such as targeting SHP2, which of course links RTKs to downstream RAS kinase, or even targeting downstream pathways, specifically MEK itself. And so I highlighted the combinations that we are testing clinically of ALK inhibitors with SHP2 inhibitors and ALK inhibitors with MEK inhibitors. Diagnostically, I agree, it's quite a challenge. So in fact, Joe, I would probably turn the question back to you at some point and ask you what you would recommend, you and your colleagues might recommend in terms of how we identify specific ligands that are driving resistance in these cases.
Right. Thanks, Alice. So again, if you have questions, please put them into the chat. And while we're waiting for those to come in, I have a question for Dr. Heist. So as we've seen now with other targeted therapy paradigms, and now with MET, we're having the luxury of having multiple therapeutic options to choose from. In your view, is there a clear first line choice for MET, and how much would you factor in that increased response rate in the first line setting, say for capmatinib, that's not really shared with the other MET TKIs?
Yeah, Erin, I think that's a great question. Yeah, as you mentioned, the capmatinib study in the first line setting showed a response rate of 68%, which has not been recapitulated by any other drug. If you look at the tepotinib data, they did break down at the latest data sharing at World Lung, their first line and subsequent line settings, and the response rates in both those settings remain at around 44%. There is a expansion cohort in the capmatinib study looking at a larger population of first line patients, and I think if that holds up, and that should be reported soon, that in my mind, capmatinib with a higher response rate would be my preferred agent in the first line setting. It has CNS activity, as you know, and with the highest reported response rate, I think you would go with what has the best activity in the first line setting.
And we have a couple more questions for you about MET as well. This is from Andy Brown, who asks whether there is any benefit to sequential use of MET TKIs similar to, say, ALK TKIs as resistance arises, and whether how often MET amp is identified in the upfront biomarker testing in the clinic.
So first, to the question of sequential inhibitors, this was a study that we actually did. We did a relatively small investigator-initiated trial. This was recently reported by Dr. Dagogo Jack from MGH, where we looked at capmatinib in people with MET skipping or MET amplification who had prior MET inhibitors. And to really ask that question of, can the sequential use of MET inhibitors add benefit here, similar to what we've seen historically with ALK? In that study, there were about 20 patients or so. Most of the patients had had crizotinib as their previous MET inhibitor, just because that was what was available at the time. And what we saw was an overall response rate of around 10%. There was some more stable disease, but overall the response rates are not what you would hope to see or would be kind of reassured to really feel like you can sequence the type 1 inhibitors one after the other. We did see some activity in cases where there was CNS progression after crizotinib. Capmatinib and tepotinib, I think both are better CNS penetrant than crizotinib is. And we saw some responses in a setting where there had been a long kind of interval without MET inhibition on the patient. But I would not rely on sequential type 1 inhibitors as a great treatment strategy in this setting. I think the jury is still a little bit out on type 2 because some of the resistance mechanisms that we see in secondary mutations in MET should be targetable by type 2 inhibitors. The question always becomes, can you get the kind of dosage that you need in terms of hitting the target with tolerable AEs with the type 2 inhibitors? But I think sequential type 1s probably are not going to get you much bang for the buck there. To the second question of MET amplification, how often we see that, the true de novo MET amplification, high-level MET amplification with a high MET to CEP7 ratio as a sole oncogenic driver event is quite rare. So we see that probably 1% or less of the time. MET amplification happens as a resistance mechanism to other targets, and we see that with more frequency, to be honest, I think, than true de novo single amplification as the driver event.
And then in terms of MET amplification upfront with other oncogenic drivers, do you see that commonly?
We've seen it more in the setting of resistance to targeted therapy. So we've seen it both in the EGFR setting and the ALK setting, and it seems to occur as a bypass mechanism of resistance. I would say we see that acquired resistance happening more than we see that as kind of right from the beginning, seeing both, for example, EGFR and MET amp. It seems to be a response to the selective pressure of targeted therapy more often than it is as a dual oncogenic driving event right after that.
And then we have a couple questions, Dr. Heist, about testing and insurance coverage. One question is whether you recommend testing DNA or RNA by NGS to look for exon 14 skipping mutations and amplification. And then a somewhat related question about assuming you see MET amp with, even though MET exon 14 or MET TKIs are not approved for MET amp, they're approved for exon 14, would insurance cover that a MET TKI if you see amp but not the skipping mutation?
So to the first question, here with our internal assay, we are looking at tumor genomics both with DNA and RNA, and I would say that for the MET skipping, we're really looking on the RNA assay, and the event that we're looking for, the deletion of exon 14, is something that can be captured on the RNA assay quite nicely. We're also looking for the various gene rearrangements that we know occur in lung cancers on the fusion assay, and I think that's kind of a very nice, reliable end product. You're seeing the end product of the change that you want to see, that you're looking for. There's a sense that the RNA can pick up a little bit more than just DNA for MET skipping in particular. As I think people who are asking these questions know, there are multiple DNA changes that can lead to the skipping event. And the RNA assay is just a little bit more, I suppose you would say, all-inclusive in terms of picking up the fact that you actually have the exon 14 deleted. So we rely a lot on the RNA assay here. There are NGS assays which are available, which do pick these up, and I think are quite reliable in terms of their positive predictive value. I think you can probably pick up a few more with an RNA-based assay. To the other question of insurance, so I would say that I have used the various MET inhibitors in the setting of MET gene amplification. It requires some jumping through of hoops with insurance and writing letters of medical necessity, but we have been able to do that. So that's something that, as an avenue, is available for patients.
Thanks, Becca. We have a question for Dr. Shah. The question is, what are the side effects of different ALK inhibitors? And I'll add to that sort of in the context of the debate about alectinib versus lorlatinib in the first line setting. to what extent do potential side effects play into, to balance out the question about, duration of response or progression-free survival? What role does that play in terms of the selection of one versus the other?
Yeah, sure. Actually, Erin, if I could first maybe amplify on the MET amp question, just because I wanted to emphasize what Becca said around how we do see MET amplification in the setting of resistance. And Becca mentioned EGFR and ALK, and I would say we see it ALK, ROS, RET, other oncogene-driven cancers. And in ALK in particular, we definitely see MET amplification as a bypass mechanism in probably around 15 up to 20% of our patients who are relapsing on ALK TKIs. And as I mentioned in my talk, I suspect that when we use these really potent pan-inhibitory ALK TKIs like lorlatinib frontline, I think we will also select out quite a bit of these bypass mechanisms, including MET amplification. So really important bypass mechanism, and that one that we can therapeutically target with MET inhibitors. So, but back to your main question around side effects. So this is a very important question. The ALK inhibitors are very well tolerated in general, but of course they all come with side effects. And I would say, interestingly, lorlatinib has kind of a unique set of side effects that do distinguish it from other earlier generation ALK inhibitors. So the most common side effects that have been reported with lorlatinib include hyperlipidemia. So this is increased cholesterol and sometimes increased triglycerides. Also peripheral neuropathy, edema, weight gain, and then notably neurocognitive side effects. And that can involve, short-term memory, multitasking, mood lability, those types. They tend to be mild, but I would say the neurocognitive and also the hyperlipidemia for lorlatinib, those two in particular really stand out as different from other ALK TKIs. Alectinib, I should note, does have side effects. So it is well tolerated, but we definitely have some common side effects with alectinib, including fatigue, edema, weight gain, constipation, and sun sensitivity. So again, all of the ALK inhibitors do come with some side effects. And I would say overall, the data suggests that alectinib in general is probably a little easier than lorlatinib. And with lorlatinib, I would say the side effects that folks struggle the most with probably do have to do not so much with the hyperlipidemia, which really is a laboratory finding and one that can be very easily managed with medications, but they struggle more with the neurocognitive side effects and sometimes the edema and neuropathy. Now, my experience has been that for lorlatinib, all of these side effects are very dose dependent. And so I think the key to really managing lorlatinib side effects is to understand what they are and to counsel patients. And really, if any of these side effects become at all challenging, it's very easy to simply hold the medication. In a couple of days, the side effects resolve, and then resume at a lower dose. And I have found that over the years, that has been incredibly helpful. It has allowed patients to basically be on a dose of lorlatinib, which they can find tolerable. And so as a result, many of my patients, once they have found that optimal dose for them, will feel like the drug is quite manageable, in many cases, even a little bit easier than some of the other ALK inhibitors. So I do think lorlatinib requires more management, requires more knowledge of the side effects, but I found it can be quite well managed. I would also point out in the phase 3 study of lorlatinib that I discussed, the CROWN study comparing lorlatinib versus crizotinib, that lorlatinib did appear to have more toxicity, grade 3-4 toxicities, but over half of those grade 3-4 toxicities were related to elevated lipids or triglycerides, which again, usually can be very easily managed. And I would note that the discontinuation rate of lorlatinib versus crizotinib, it was lower with lorlatinib than crizotinib. Again, highlighting that this drug overall can be managed. And then the last thing I would note from that CROWN phase 3 study is that when we looked at patient-reported outcomes, that there was a greater improvement in global quality of life relative to baseline in the lorlatinib versus crizotinib patients. So again, I think it highlights that, yes, there are some side effects, but for most patients, they can be managed. The last thing I would note, Erin, is that, the standard dose of lorlatinib is 100 milligrams a day, and that was established in our phase 1 dose escalation study. However, in my experience, I would say most patients really have difficulty tolerating 100 milligrams. And we saw in the phase 1 that lower doses actually were able to fully inhibit the target, including some of those refractory resistance mutations. And so even doses like 75 are very effective, both clinically and in preclinical studies. And so I actually usually start my patients at a dose that's lower than the recommended dose, because I think that's for me the best balance of efficacy and side effects.
Thank you, Dr. Shah. And we have a question for Dr. Heist about the MET antibody drug conjugates and what your thoughts are in terms of resistance to those.
Yeah, I think that's a great question. As you know, there are MET antibodies and MET antibody drug conjugates that are being studied. I think what we've seen from the resistance studies for the TKI so far have already shown that there are off-target pathways of resistance, specifically RAS, MAP kinase, ERK pathways, and ERB pathways, which seem to get activated. So I would think that those similar pathways may get activated even with ADCs or antibodies. The jury's still out on this, obviously, and there's still work to be done here because we haven't really fully investigated this setting yet, and we're only now starting to get some activity data. But that will be an area of high interest as we go down the road.
We have a question for both of you regarding the use of plasma liquid biopsy, looking at ctDNA, looking both at ALK and MET exon 14, and where do you think that's most useful in relation to tissue biopsies?
Yeah, I can start. I mean, in the upfront setting, I think we primarily rely on tissue diagnosis of rearrangements such as ALK and ROS1. And of course, for ALK as an example, we have a number of different ways to diagnose ALK rearrangements in tissue that are very, very effective, including IHC and FISH. And so I typically rely on tissue. However, over the years, there have been patients who, for various reasons, did not have sufficient tissue for testing and really couldn't undergo another biopsy. And so in that case, yes, we can use a liquid biopsy or ctDNA at the time of diagnosis to identify targetable alterations, including ALK. There are atypical ALK rearrangements, and those will oftentimes just not be picked up in ctDNA. And so that's why my preference is to rely on tissue, at least in the upfront setting. But in the setting of resistance, where we are tracking mutations, such as the ones I mentioned during my talk, those often can be tracked quite easily in liquid biopsies. And so at the time of resistance, I would say most of my patients will often opt for liquid biopsy, so ctDNA. And in the setting of having an uninformative or negative ctDNA, then we will often then proceed with a tissue biopsy for further information on resistance.
Yeah, I agree. We rely still a lot on tissue, especially for that initial diagnosis. We have started to use liquid more and more, even initially, simply because the range of alterations you actually need to know about in the first line setting as you're trying to make decisions about treatment seems to be ever expanding. And the liquid does give you a fast look at that. I think the cautions we have are that there are some tumors that don't seem to shed that much ctDNA. So if there's relatively limited volume of metastatic disease, if it's primarily in the lungs or in the CNS, those are situations where we can't always rely on the liquid to tell us what we need to know. And a negative liquid biopsy doesn't mean there's nothing. It just means that it may not have been a very actively shedding tumor. For resistance, we use the liquids a lot, as Alice was saying as well. I think the one thing you don't catch on the liquids is histologic transformation, and we do want to know about that. For some of our patients, sometimes you get a sense of that just on the pace of the disease and what's happening in terms of the tumor extent. But there's still definitely a role for tumor biopsies, particularly in looking for and defining histologic transformation as resistance.
Thanks. We're almost out of time, but I think we have time for one more question. So this question, I guess, could be for both of you. It's a general question about the time frame at which point resistance is observed, and I'll add to that: is there anything known about the time frame of when different classes of resistance may arise, say on target versus off target?
Yeah, so I mean, for ALK TKIs, ROS1 TKIs, I mean, it's a huge range in terms of when resistance may develop. In general, though, as I pointed out during my talk, the time frame is longer when we use more potent and CNS penetrant TKIs in the frontline setting. What I've been really impressed by over the years is how how huge the spectrum is. Whereas, we can have a patient who relapses within a few weeks of starting on a TKI. And then I have some patients who have been on crizotinib for now over 12 years as their first TKI. So it's a huge range, but there are general sort of ballpark estimates. And that's basically captured in a lot of in the trial data. Erin, in terms of your question around mechanism, again, I haven't figured this one out. Maybe you have, but I have had patients who have relapsed on the early side, and we've often wondered, are those patients the ones who have a specific class of mechanism of resistance? And actually what I've seen is that they can have almost anything. They can have MET amplification arise very early and it drives relapse at 2 months, or they can have had G1202R emerge very early and probably was a pre-existing subclone that was there from the beginning that then just grew out. So I have not been able to correlate clinically the type of resistance with sort of the duration of response. Becca?
Yeah, and I would agree with that. And, for the MET skipping, I would say in general, there is that wide range that you can see. If you look at the MET skipping time to progressions, those are shorter in general than what you see for example, ALK and ROS. So I think there are differences between the different types of alterations. But within the alteration, there is a wide range, with some people having really prolonged time before progression and some people progressing relatively quickly. But between the different classes of alterations, there are probably some ranges which are quite defined and different.
Thanks. I'd like to thank Dr. Shaw and Dr. Heist for terrific talks and terrific discussion. I'd like to thank Dr. Chabner for hosting us, and I'd like to thank all of you who are watching for your questions and for your participation. And I hope everybody has a terrific day.
Thank you.
Thank you.